You can hide this if you want to. Sure. Okay.
Lower relatively that because it's kind of too high. Okay, hello everybody. For those of you who don't know me, my name is João Jun. I'm a professor from the University of Lisbon on a sabbatical leave here at Barcelona. And today we have a visitor from Portugal as well, Afonso Mota. He was my student in the early years of his grau, his bachelor degree in biology. So he was the first biology student who decided to learn some astrophysics in order to do something that he would like to do in the field of astrobiology. So he kind of a pioneer making this connection between biology and astrophysics. And then he went on to Naples, to Italy, for his master's degree. He has a master's degree in biology of extreme environments. And for his thesis in master's thesis, his project was on exoplanet habitability in collaboration with the German Aerospace Center in Cologne, Germany. Now he's about to start his PhD at the University of Portugal with Professor Nuno Santos and as co-supervisor and also supervisor from France. And he will work on coupling astrophysical models and microbiology experiments to determine exoplanet habitability. Now he's here to discuss possible collaborations in the field of astrobiology with Octavi and Andrea. Yeah, yes. Who are very interested in astrobiology. They have been keeping alive a little bit of astrobiology here at the University of Barcelona. It's an interdisciplinary field, and in my own belief, interdisciplinarity is a really important thing that will strengthen any field of research and knowledge. So after that, I'll give the word to Afonso.
Okay, thank you, Professor João, and I'd also like to thank Octavi and Andrea for the invitation and everyone for being here today. As Professor João mentioned, I am from biology, but I work on astrobiology currently. Is it the same? Okay. And specifically in the bridge between astrophysics and biology or microbiology in the study of exoplanet habitability. And that's what I'm here to talk to you about today. Why? Okay.
To change the mic cable
To another one?
Yeah.
Sure.
There is another word.
I think it's fine. Yeah. Okay. So as I was saying, the bridge between astrophysics and biology, and I'll tell you this in general terms, but I'm also going to tell you about my specific research, which I did at the German Aerospace Center in Cologne while I was doing my master's degree in the University of Naples in Italy, and I'm currently, as Professor João said, associated to the Portuguese Institute of Astrophysics and Space Sciences. But first, just a little bit about me. So as was already mentioned, I have a bachelor's in biology, and I also did a minor in physics because ever since the beginning when I started my journey, I really wanted to grasp the concepts not only of the biology side of astrobiology, but also the physical side and the astronomy side of studying planets and the universe in general. I then went on to do my master's degree in biology of extreme environments with a specific curriculum on astrobiology in Naples. And now I will start soon my PhD in astronomy at the University of Porto. So it's got quite a, I guess, amazing run so far, but that's exactly what I'm looking for. It's different expertise in all of these fields. Just as a brief outline of my talk, I'll first give you a general overview of astrobiology and exoplanet science, both from the biology side and from the astrophysical side. And then I'll dive a bit deeper into my research, which was focused on M-dwarf habitability, as you'll see. Starting from the first topic then, I'll go with a very broad question, which I'm sure we've all wondered at one time or another, which is, are we alone? Are we alone in the universe? Are we the only planet that has life in the universe? Or even if we aren't, are we the only intelligent species in the universe? These are all really interesting questions about the cosmos that have plagued humanity for millions, not but thousands of years, even before we started looking at the stars from a more scientific perspective. And I really like the quote from Arthur C. Clarke, who says that two possibilities exist. Either we are alone in the universe or we are not. And both of these are equally terrifying because whichever the answer is, it's pretty scary to think that we are alone in the universe and there's nothing else out there. But it's also kind of scary to think that something is out there, then what is it? And how can we interact with it and see what it actually is? But I also like the substitution from Charles Cockell, who's a prominent researcher in astrobiology, who has that these possibilities are both equally exciting. And I agree because it is scary, that's true, but it's also incredibly exciting to think that there may be something else out there in the universe. And whichever the answer is, yes or no, it would profoundly change the way that we look upon ourselves and in the cosmos as well. And this is kind of the basis for the question of astrobiology. And as a science in itself, astrobiology studies the origin, evolution, and distribution of life in the universe. So it's quite broad. And not only does it research the fundamental nature of life on Earth, so how did life originate? What is life made of? And how can you even define life? Because that's a very important point. And of course, the possibility of life in other places, not only in our solar system, but beyond. And it's kind of obvious that it's a very interdisciplinary field, just by its name alone, astrobiology. It includes fields like astronomy and biology, the study of life and the study of planets, but also chemistry. When you think about what we are made of, we're made of molecules. So how do cells interact chemically, and what are cells actually made of? Geology, of course, for the study of planetary sciences. But you also have other fields which are really important to the development of the field, like engineering and even philosophy, among others. Just a quick history lesson. So the first mentions of astrobiology, or exobiology as it was called at the time, date around to the 1960s. But as you can see, it didn't get much traction for about 20 years or more. And after that, there was kind of a boom in the study of life outside of Earth and the idea of the possibility of life outside of Earth. But it's still a relatively recent field that has, of course, gained a lot of traction in the last few years with improving technology and the knowledge we now have about more planets and their characteristics. And even in our solar system, discovering that Mars used to have water, perhaps it could have had life, that really brought a lot of attention to the field. But it's still relatively recent, of course. And it was pioneered by some of the greatest scientists of our generation, like Carl Sagan, Frank Drake, and of course, myself. As for exoplanet astrobiology, so this is the exoplanet science in general. It's a subfield of astrobiology where you study planets outside of the solar system. The first ones were discovered in the 1990s, but since then there's been a near exponential discovery of new planets. And this has led to our understanding that there are quite a lot of different worlds out there than ours. This is just an image of or a video of a star with some planets around it. These are giant planets, not rocky planets, but we can do some of this direct imaging. But most of the time, we're studying the planets indirectly. So what happens when they pass in front of their star, or how do they affect the gravitational effects of their star, and how can we detect that? And so we don't have a lot of detailed information about many of these planets. And so how can we study that? At the same time, we know of more than 5,500 exoplanets, but a lot of them, we only know that they're rocks, or maybe not even rocks, maybe bundles of gas in space. So how can we actually translate that knowledge into understanding how they may harbor life or not? And for that, of course, we have to define what it means for a planet or a body to be habitable. And so there are two concepts that are generally spoken about. You have habitability and habitable zone. The first one is quite consensual. Habitability is a measure of a planet or natural satellite's potential to develop and maintain environments hospitable to life. I think this is quite consensual within the community. But the second one raises a bit more doubts. And it's a concept that dates around in the 1990s, as well, around the time when we started discovering exoplanets. And it mentions a habitable zone, which is the circumstellar region around the star, where an Earth-like terrestrial planet with a suitable atmosphere can host liquid water on its surface. And this has been a key concept in astrobiology so far, but it has several issues. When we look at a habitable zone, it's more or less something like this, where you have an inner limit where planets after that limit would be too warm to contain liquid water on their surface and therefore life, and planets after the outer barrier would be too cold. And this has been the key metric for astronomers and astrobiologists to look at planets and see where they are situated in their orbits and determine whether or not they may be habitable. But it's been questioned whether this is the best approach, because there are several counterexamples nowadays. One of the main ones is, what about moons? Even in our solar system, we have a lot of moons like Europa and Enceladus that are very, very well known to have subsurface liquid water oceans that may or may not have the conditions suitable for life. Especially Enceladus, with the Cassini mission, has been extensively studied and has very promising results about not only the availability of water, but nutrients and other energy sources that could allow for the development of life. And these are moons that are way outside of the habitable zone. They are not Earth-like planets, and they don't have liquid water on the surface, and yet they are considered very likely habitable. You also have the example of planetary subsurfaces. So the surface may be habitable on a lot of planets, but below the surface, for example, on Mars, you may have pockets of liquid water or otherwise habitable conditions, even though in this case Mars and many other planets are outside of the habitable zone. And finally, you have different classes of planets that have been discovered in the last years and decades. This example that I put here is of Haitian planets, which actually have been discussed a lot recently, which are non-terrestrial planets that have water oceans, a thick hydrogen atmosphere. It was clicking for some reason, sorry. And they have a thick hydrogen atmosphere. And these, of course, are not Earth-like planets. And their so-called habitable zone, where they can have actually this liquid water on their surface, is wider and different than the traditional Earth-like habitable zone. And there's another important factor here for anyone who is from biology. It's extremophiles, or the so-called extreme-loving organisms. And these are generally microorganisms, so bacteria or fungi, that inhabit places that are very extreme, at least from an anthropogenic point of view, and that were thought to not contain life until relatively recently in some cases. Just as a few examples, you have hyperacidic lakes and volcanoes with, of course, very acidic and hot conditions. You have deep sea hydrothermal vents with, once again, very high temperatures, extremely high pressure. You have soda lakes that Andrea studies that are extremely basic. It's almost like swimming in bleach, but a lot Finally, yes. We should be. Nice. Okay. As I was saying, extremophiles, blah, blah, blah. Let's keep going. And just these two examples can show you how astrobiology is uniquely interdisciplinary as a field. And you can learn a lot from looking at the other subfields within astrobiology. So the examples I gave you about astrophysics, the different types of planets, the different types of orbits, of conditions that there are, really can inform us about what constitutes a possibly habitable planet and even what kinds of conditions there are in the universe. And when you look at the biology side, you can just look at what organisms exist and how they may work and react to being in these environments. So especially modern day astrobiology is inherently multidisciplinary. And of course, then it necessitates a lot of learning from each other and collaboration. But this is all well and good in theory, but in practice, it's not that well applied in many cases. I'll give you my example. I was a master student just studying my master degree. I had some experience with astrophysics from my minor, but I was mostly a biologist. And part of my research was to study how X-rays from red dwarf stars, which I'll talk about in a little bit, affected life. And I wanted to know what dose of X-rays I should irradiate my organisms with. Easy, right? So I wanted about M-dwarfs, which are the type of stars, and I wanted to work with X-ray flares. And this is the kind of plots, I guess, that that appeared. And of course, to an astrophysicist, maybe this makes some sense. And but for someone with a background in biology, this is quite overwhelming at first, and it's hard to make sense of what it actually is important here. But above all, it feels like a language barrier, like scientists from different fields write in slightly different ways, and it's kind of hard to decode that. And that's more or less what this looked like to me. It was complete gibberish. And although collaboration and interdisciplinary teams mitigate some of these problems, I believe that an integrative approach should be developed where you can bridge the gaps between subfields and habitability research and also increase the reproducibility of your research. Because if I want to do the same as these astrophysicists did and maybe remake their models or adapt them for my experiments, it's impossible for me to do so if I can't really understand anything. And of course, you have to have some kind of specialty and interdisciplinary side. But I think a balance should be reached, and we're still a bit far away from actually achieving that. Now on to my actual research that I mentioned. I worked with M-dwarf stars and the habitability of planets around these stars. And these are also called red dwarfs, and they're the smallest, coldest, and most common stars in the universe. And being the most common stars, it's about 76% of stars, at least in the galaxy. They're quite interesting for astrobiologists because we've detected a lot of planets around them, and especially for rocky planets, it's easier to study them, at least right now, around this type of star. But they have some issues. One of them, and the most well-discussed one, is the production of powerful flares, which are energetic outbursts that release a lot of radiation like ultraviolet and X-rays that is really detrimental to life and may even sterilize a planet which otherwise may be habitable. I worked on a star system called TRAPPIST-1 and another one that I'll tell you about. This is a scheme of TRAPPIST-1, and I worked specifically with 3 planets inside this habitable zone, planets D, E, and F. And I know I was just saying how the habitable zone is not quite the best concept for habitability, but it is still a useful tool for researchers to guide their experiments and their research, as long as we make sure to clarify that this does not mean that planet G cannot be habitable in any way whatsoever. It just means that in this case, I'm focusing on these ones because if they are Earth-like, they have the highest likelihood of being habitable in this orbit range. And one of the biggest effects I studied is the one I already mentioned about radiation. And one of the traits of red dwarf planets is, since these stars are much colder and smaller, for planets to be in the habitable zone and receive enough energy to have possibility of life, they have to orbit much closer than for warmer stars. And if they're much closer, they also receive the brunt of the flares and since these flares are even more powerful than stars like the sun, this high radiation environment can really be an inhibitor for the origin and development of life on such a planet. And so my objective overall was to study how habitable these M-dwarf exoplanets could be, keeping in mind all the problems or potential problems that life faces as it originates there. And I wanted to take an interdisciplinary approach by first modeling the conditions on these exoplanets. I mainly focused on temperature and radiation and check whether these may be conducive to life. But I also wanted to characterize its habitability and on a more practical sense by taking the results from my models and what I think is our best guess of what these planets could look like and seeing whether Earth microbes could survive there. So taking actual organisms and doing experiments on them. And of course, as I already mentioned, I took an interdisciplinary and modular approach because I wanted this to be reproducible and for future researchers to be able to take my models and apply them in a more or less straightforward way, bridging the gap between astrophysics and microbiology and a bit of biochemistry. The outline of my project was for me to first select my star systems, which were TRAPPIST-1 that I already mentioned, and Proxima Centauri. I worked with planets B and D, which are the rocky planets of this system. And as I said, this was developed at the German Aerospace Center under the supervision of Marta Curtzel and co-supervision of Professor Nuno Santos in Portugal and Professor Marco Moraci in Naples. And I studied a few conditions, but as I said, I mainly focused on the surface temperature and radiation of these environments. And then I exposed the microbes and analyzed the results. I'll start with the surface temperature modeling that I did. And I'll mention the example of Mars and how that differs from how we study exoplanets. So when you're studying the daily surface temperature of Mars, for example, this is a model for the distribution of that factor. And this is based on data from decades of space probes, satellites, and observations. And so we have quite a detailed knowledge of how temperature variations work on Mars and how its atmosphere circulates the energy and all of that. And you can do the same for exoplanets. For example, this is one for Proxima Centauri b, assuming a few conditions. For example, assuming the synchronous rotation of the planet, which is when the planet always has the same face turned towards the star. And I'll talk a bit more about that in a little bit. And also assuming that its atmosphere is just 1 bar of CO2, which of course is not realistic at all, but it's still an interesting supposition. But you can also assume 50 other conditions, and you'll get 50 different models. So this doesn't really represent what we think Proxima b looks like. It's just the different possibilities that there are. And although this is, of course, extremely useful, especially from an astrophysical point of view, when you're trying to design a biological experiment, you want a little bit more flexibility and ease of use to be able to have a better developed pipeline, shall I say. So what I did was I developed a simpler model, a 0D model of a 1-layer atmosphere, considering, so based on the equilibrium temperature, I then added a few factors based on previous research, where I considered, of course, the flux that reaches the planet, the energy flux from the star, its albedo or its reflectivity, the greenhouse effect that its atmosphere has, its energy distribution efficiency, which has to do with whether it's synchronously rotating, so if it's tidally locked or not, and the number of atmospheric layers, which is done to simulate very dense atmospheres like the one on Venus. So this is quite a simple model. It's just one equation, but the advantage is that it is still extremely accurate for solar system planets, and it may be accurate to some degree outside of it. But above all, it's extremely flexible. And the output of the model is a surface temperature matrix like this one here. So this is for Proxima Centauri b, where depending on its energy distribution efficiency, albedo, and greenhouse effect, you have the predicted temperature values. And from this, you can then explore different scenarios like they did before in the other paper where they had a few discrete scenarios. Here, you can explore it as you wish. So for example, what if Proxima b is tidally locked or synchronously rotating, just like our moon is to the Earth? When planets or bodies orbit very close to each other. Sometimes there's this effect where you always have the same phase turns towards the star. And in that case, the day side is much warmer than the night side, unless there is a very efficient energy circulation, which is this energy distribution factor that I mentioned. So if it's tidally locked, then that factor will be very low. So it's half the planet, so 0.5 gets all of the energy. And in that case, we can then assume, for example, okay, what if what if Proxima b has Mars-like conditions? So it has a very thin atmosphere and otherwise Mars-like conditions. Then it's predicted day side temperature, so the temperature on the side that receives light, is about 3 degrees Celsius, which is really relevant information, not only to then plan your experiments, but that tells us that it could be possible that under certain conditions, Mars could have life on the surface or perhaps liquid water, depending on if the atmospheric pressure was a bit higher. And if we assume Earth-like conditions, then we see that it's actually quite good. A bit colder in here, but in general, it would be likely that you would have many areas of the planet on its day side that would have great temperatures suitable for the presence of life and of water. And so I started building this hypothetical exoplanet scenario using Proxima b as an example. As I mentioned, a tidally locked planet in the habitable zone of its M-dwarf star. And we just saw that the temperature could be conducive to the presence of surface liquid water with an atmosphere that was thicker than Mars, but not necessarily as dense as Earth's and much less dense even. And of course, this is to some extent speculation, but it's still relevant because the atmospheres of rocky exoplanets are mostly unknown currently, and we will develop techniques and methods with new telescopes and more information we get to understand how these environments actually are. But to build the experiments and to be able to justify what kind of conditions we are using in the lab is really important in this scenario of bridging the gap between the two fields. I then wanted to look at how much radiation reached the flares, reached the planets during the flares. Once again, just to remind you, flares are these very strong outburst events, which actually the sun is currently amid one of these outbursts, and that's why we've been getting great auroras all over the world, because we have a magnetic field or the Earth has a magnetic field that protects us. But if you don't, all of this radiation can reach the surface and, well, fry you. And both of these stars produce flares. Proxima b, Proxima Centauri, sorry, in orange, produces a bit stronger flares than TRAPPIST. And here you have the data for what I modeled would be the ultraviolet or X-ray radiation that would reach each of their planets. So as the dots here, as you can see in Proxima, they will receive higher amounts of radiation. And just as a comparison, because these values don't really mean anything without a comparison. So an average flare, the UV fluence, so how much ultraviolet light would get to Proxima Centauri b at the top of its atmosphere, if it has one, would be around 377 joules per square meter. And the average bacteria, for example, survives about 16 joules per square meter. So this is extremely high. And a similar event in the solar system would reach the Earth, even not counting magnetic fields and atmospheres, with a UV dose of not even 1 watt per square meter, which is extremely low.
This is the joule per square
Yes, and then I put so here I'm assuming an average duration of the flare, and here I'm not. So this, yes, it's of course it's not the same. So if you have a very long flare, this could grow a lot, but the value is quite a lot lower than the one on Earth. 60 seconds. 60-second flare here. That's what I'm assuming. The duration I'm assuming here is 60 seconds per joule. So this would be about 60 watts per square meter. So if 60 joules per square meter if we're talking about a 60-second flare once again. Okay. Well, it's about six times different, or 5.
Biology is adaptable.
Well, as I'll show, more or less depends on what organisms you consider. And in this case, the higher values, you get an exponential effect of depth of the organisms. But I'll get to that in a little bit. And once again, a super flare on Proxima b, so this is a much stronger flare, could blast up to 130,000 joules per square meter onto the closest planet, so planet D here, over a few hours. So here for the super flare, I'm assuming 2 hours of flare activity, which is, let's say, worst case scenario of radiation. And of course, this would be quite bad for anything on the surface. But I also wanted to know, okay, this is what reaches the top of the atmosphere, but what if, what actually reaches the surface if we assume that there's a Mars-like atmosphere on the planet? And what I found was that knowing the distribution of the flares, most of the lower wavelengths of UV are completely absorbed by the Mars-like atmosphere. So this is a spectrum of Mars-like UV transmission for 3 different scenarios: minimum transmission, intermediate, and maximum. The difference between these is the amount of dust in the air and the angle of incidence of the photons, of the UV photons. And I worked always with the maximum transmittance scenario, just once again looking at a worst case scenario of the highest amount of radiation that would get to the surface. But the good news is that most of the lower, from the higher frequencies and lower wavelengths are blocked by the atmosphere, and these are the ones that are most dangerous for organisms, any organism in general. Then I wanted to look at X-rays, and this is where we kind of get a moment like I was describing before of a bit of confusion on the biology perspective, because unlike for UV, where the dose is measured by watts or joules per square meter, X-rays are measured in terms of the absorbed dose or joules per kilogram, which is because in a lot of cases, especially higher energy X-rays, they can go through a tissue or a material and not be absorbed. So what we actually count is the dose that is absorbed. And this, of course, is better to describe what the organism will actually face in terms of a radiation amount, but it's greatly dependent on the wavelength, and there's not really an easy conversion from one unit to another. And the flares, when we talk about flares from stars, energy factors are always given in joules or erg, which is an equivalent unit of energy. And so I had to develop a way to convert the irradiance into absorbed dose. So I get the flare X-ray energy, so let's say 10 to the power of 30 ergs, and then I have to assume the power law distribution for the flare, which means that all the flares you know of have this kind of distribution where you have many low energy X-ray photons. This is of course for the X-ray band, many low energy X-ray photons, and very few high energy X-ray photons. And this allowed me to then use a conversion factor, which is the absorption coefficient, to calculate the dose at the top of the atmosphere, and then finally model the attenuation from a Mars-like atmosphere, which was what I was studying, as I mentioned, and then get the surface dose. And what we get here is, once again, that, so this is the my fit. It was an easy, a simple fit of the model of the observations that have been done on the Martian atmosphere for its X-ray transmission. And what I saw is that, once again, most low energy photons, or almost all of them, will be blocked by a Mars-like atmosphere. And as I previously mentioned, the vast majority of the photons that are emitted in the flares are on the X-ray band are low energy. So only the higher energy ones pass, and those are very few. So the actual dose on the surface would be almost negligible. And just as a recap so far, in this Proxima B scenario under a Mars-like atmosphere, essentially no X-rays would reach the surface. The dose would be negligible, but some UV radiation would, and it could be detrimental to life. And you, of course, have to take into account constant cosmic ray and proton flux, which I did not work with, but is definitely a factor in survivability, of course. And so far, these conditions sound quite habitable in terms of temperature, in terms of radiation environment. It could be survivable for many organisms on Earth. But the sad thing, shall we say, is that under these very heavy radiation environments, without a powerful magnetic field, the photodissociation of molecules, so molecules are hit by X-ray and the UV light, and they're broken apart, and then they're lost into space. And after billions of years of planetary evolution, it's very likely that you get planets that were stripped away of their atmospheres, leaving the radiation to reach the surface unimpeded. This is what, for example, happened on Mars, that lost its atmosphere over time and has now a very residual atmosphere. And this was in a scenario that is much less intense on radiation than M-dwarfs, especially, both constantly and during the flare events. And of course, these high radiation doses on the surface may sterilize it completely, but a lot of organisms on Earth live in the subsurface, especially when you're talking about bacteria. There are some that can live hundreds of meters or more even below the surface. And so I wanted to see how far these X-rays could penetrate the surface, since these were by far the highest danger to the organisms if there was no atmosphere. And what I found is that using the characteristics of either water or Mars-like soil, the dose would rapidly decrease in just a few centimeters or millimeters of soil or even water. Most organisms would be able to survive, from the strongest and most hardy, like the Dinococcus aeluminans, but even the weakest ones, shall we say, like E. coli, could survive under maybe a few millimeters of soil. So if life is able to originate when the planet has more suitable conditions, it will then certainly migrate into the niches underground and possibly be able to survive there long term. But I also wanted to know whether organisms could survive on the surface, because on Earth we have many examples of radiation-resistant organisms that employ many strategies to survive. And one of those, and one of the most important ones, is the production of pigments, which allow the absorption and protection of the organisms from very high radiation environments. And I particularly wanted to study melanin, which is one of the most common pigments on Earth. We produce melanin. That's what we use to protect ourselves from the ultraviolet rays of the sun. And recently, it has been proposed that melanin could have been delivered exogenously through meteorites early after the Earth's formation. So, as you know, some meteorites they contained a lot of water, amino acids, and sugars, for instance, that were really important for giving the chemical product for the origin of life. But they also brought insoluble organic matter that was very chemically similar to melanin and was rapidly converted into melanin with UV light. And especially since melanin is widespread in the web of life, so a lot of organisms from different life groups have melanin and produce it, from the simplest cells that have melanin to us, the most complex organisms. There is melanin in a lot of them, and this indicates that it could have originated very early in the evolution of life and then have been maintained by a lot of those organisms, and therefore it could have had a role on the origin and proliferation of life itself on Earth, and perhaps the same could be true elsewhere. Now, before I go into the experimental section, I'll just recap what I've mentioned so far, that I developed simple models, but versatile ones for exoplanet environments. And using those, I estimated that for a Mars-like atmosphere, planets like Proxima Centauri b and TRAPPIST-1e would have low radiation environments, but very high radiation doses without an atmosphere, but life could be possible at the subsurface. And now I wanted to see whether terrestrial life could survive on these exoplanets. And I wanted to test microbial survival to particularly the flare radiation, which looked to be one of the most constricting factors to their survival. And I also wanted to explore the role of melanin in the radiation shielding of these microbes, and whether the presence of melanin could be the difference between a sterile planet and one where life is able to proliferate despite of these strong flares. To do so, I had to first select a model organism. And for those of you who don't know, a model organism is a species that has been generally employed for a while and is extensively studied to understand a particular phenomenon in biology. And for example, you have corn for agricultural studies, We've all heard of studies on mice for medicine or biotechnology, flies as well. And there are also equivalent model organisms for microbiology. And the discoveries made with these model organisms may provide insights into how other organisms may react. But of course, we have to be careful to constrain that to each organism's characteristics. I worked with fungi, in particular filamentous fungi, because that was what my supervisor worked with, but also because they are extremely resilient organisms. They are considered microorganisms because they have their individual cells. You can grow in individual cells as microorganisms, but they also form these colonies that are visible to the naked eye, are more or less this size. And where you see here, this black pigmentation, that is melanin that is present on the spore. So this is electron microscopy of a spore head, and each of these is a spore. And the spores are the most interesting and resilient parts of this organism. They are very hardy structures that are more or less the seeds that they launch into the environment to reproduce. But they're highly resistant to many conditions, one of which is radiation. And it's partly due to the high amount of melanin that gives them this characteristic black color. And they've also been studied in space. This here is a photo of a panel in the International Space Station, which has black mold, which is this particular organism. And we've also, maybe not all of us, but a lot of us have had black mold in our houses. They're very attracted by humidity, and they're extremely hard to get out of your house. If you've ever had that experience, it's not good. But yeah, they had to remove this panel and throw it into space because they couldn't get rid of the fungi. This is just a video of the germination of Aspergillus niger, which is the fungus I work with. As you can see, each of these dots are one spore, and then they start to grow these hyphae. So there's canals of continuation of their cells. And then they're joined between each other to form the final colony, which is visible to the naked eye, as I showed you. As I mentioned, Aspergillus niger was the organism I worked with, and I worked with 3 strains. Just like every organism has strains, but in microbiology they're quite common, and they're the same species but have some variability. I worked with the wild type organism, which is the standard version you'd find in the wild. Which of course has these spores that have melanin that gives it their tolerance to UV and X-rays. I also worked with 2 modified strains, 2 mutants. The first mutant was genetically engineered to excrete melanin into the environment, so it still looks the same, but when it's grown, it excretes melanin into the environment. And another strain, which is melanin deficient. That's why it looks a bit lighter, because it produces much less melanin in the spores. To measure the actual survival and growth of the fungi to the conditions I would place them on, I used two strategies. I of course irradiated the samples first, either with UV or X-rays, and then I did survival assays where you essentially verify up until what dose the organism can survive and grow these little colonies here. And I also used the oscilloscope, which is what makes these kinds of growth videos that you see. They're made by the oscilloscope, which follows the growth of the organisms over a period, in my case, 48 hours. Starting with the X-rays, I first wanted to test how well a Niger germinated after being exposed to the X-rays from a flare. And these doses here are assuming that there's no atmosphere on the planet, in general Proxima Centauri b or TRAPPIST-1e, which are considered to be the most likely to be habitable. And these doses are, and then the maximum dose here is 0.2 millimeters below the soil. So below a very shallow soil layer, you get 1,000 gray of dose. And I also did a few intermediate doses to see how the organism would react. On the left, you have the wild type strain, so the one that produces melanin normally. And on the right, the melanin mutant strain that does not produce so much melanin. And the relevant factor here that we saw is that, of course, they grow less if they're irradiated with more X-rays. But at the dose of 500 gray, the wild type was able to grow, but the mutant was not able to grow as well. This growth here is essentially probably just a standard error, whereas this one is actually a bit higher, and we can see the growth on the images. Just as a general example here, we have more growth at 1,000 gray from the wild type when compared to the mutant. And of course, this is the growth that you see without irradiation. So there's clearly a big effect even at 500 gray, but the wild type maybe was able to survive the slightly higher dose, at least grow better, perhaps due to the presence of melanin. I also wanted to look at how solubilized melanin would affect the survival and growth of this organism. What does this mean? So, as I mentioned, it was estimated that melanin would have been delivered to the early Earth. And in that case, it would be, if it was incorporated into life at the time, it would be mixed within the water solution where the protocells were, and given it additional radiation protection or other positive effects. And so I wanted to see if the melanin in the solution with the cells would give them this additional protection. And as I said, I had these two strains, and besides the wild type one, I had this mutant one, which produced and excreted the melanin to the outside environment. And I then took a sample of this liquid, and as a control, I used the wild type strain to be run in a similar liquid medium, which will produce the same organic compounds and sugars and metabolites that are excreted here, with the only difference being that there's no melanin in these samples, the control ones, but there is melanin in these ones. And I wanted to see how these different solutions would affect their survival. And what I saw was quite clear, that melanin drastically improved the spore germination, survival, and general growth. We have, first of all, here on the left side, this is without any radiation. So just at the start, these are synchronized in terms of time. And you see that the spores that were in a melanin-rich solution grew much faster and much better than the ones in a control solution. And keep in mind that the only difference here is the presence or absence of melanin. And the reason for this, most likely, is that melanin is not only good for radiation protection, but it also has a lot of other cellular roles in cellular messaging and protection against reactive oxygen species, which are essentially the oxidative stress that we even talk about as humans to try to eat something with antioxidants. That's why it's important, because it keeps your cell health higher. And so even without the radiation protection, it was really helpful. But if we look at irradiation first, starting with ultraviolet, we see three solutions here: the control solution without melanin, the melanin-rich solution, and then one that's simply seawater. So just water with the salt and nothing else. And you see here that that's the one where even at 1,000 joules per square meter, the cells die very quickly. And this is the difference here is due to the lack of any other nutrients, any other compounds that could be used to fix the DNA and to fix the cell after being irradiated. Whereas the control and melanin solution have similar survival values and in 1,000 joules per square meter. But the higher the dose, the higher the difference and the variability between these two, where at 5,000 joules per square meter, here you have about 2 or 4% survival rate, if I remember correctly, and almost half of the spores in the melanin survive. So there's quite a big difference in the protection factor of that solution. And a similar effect is seen for X-rays. So in X-rays at 500 gray, these both still survive, but at 2,000 gray, none of them survive. But the only one that survives 1,000 gray, which, as I mentioned, was just slightly below the surface, was the ones which had some protection from the melanin in their environment. And what this shows is that melanin not only has this very important role in radiation protection, but in general cell health. And if it was present in the early primordial soup of the origin of life, or at least after life was formed, it would definitely have a very beneficial value evolutionarily to be not only in the environment, but present within the cells later. Just to conclude, as I mentioned, I developed these models, these simple but versatile models, to study exoplanet conditions and habitability. And I then used them to assay the radiation environments from specifically M-dwarf exoplanets, and then study the microbial survival on these environments and the potential role of melanin, so which is also a potential biosignature that is a sign of life on another planet. And if there are planets covered with pigmented organisms, not necessarily melanin, but other ones, and there have been a lot of discussions about that recently, melanin is a potential biosignature, and if organisms with it are benefited, perhaps some planets have similar scenarios. But the take-home message and the key takeaway I'd like you to get from here is that not only did the research improve knowledge about exoplanet habitability, but above all, it helped me create modular tools for estimating the conditions on exoplanets and to inform further experiments, not only mine, but in general. And I think this approach and this perspective is really what will make the difference in astrobiology in the future on a more interdisciplinary focus, and also not only for collaboration, but for each individual researcher to know more about all of these subfields. If you're curious about my research, I recently published the preprint. I'm still waiting for the review. Been waiting for like 3 months, but anyway, the preprint is out, and that's it. You can contact me here. Also, if any of you speak Portuguese, we have a page for astrobiology science communication in Portuguese. Feel free to check it out. But otherwise, that's all I have for you. And if you have any questions, feel free to let me know.
Thank you very much for your very interesting talk. We have time for questions.
I was a bit surprised that you focused a lot on melanin, because the discussions on the origin of life, in the Earth, which is what we know. Of course, in the beginning, people talk about the role of UV from the young sun in creating massive, right, and primitive Earth atmosphere. So it seems like the question was the opposite, how to create some molecules that are essential for life by using UV. For which there was a lot more primitive Earth, because the sun was young and there was no ozone. So it was 100 times or 1,000 times more than today, right? And then we seem to be concerned about the factors of 10 or so about other planets, which for an astrophysicist, of course, and we start with the factor of 10 is like nothing. We just always find it.
Yeah, well, so I based myself on melanin, as I mentioned, because there was a study in 2021 that came out that indicated not only that melanin was delivered to the early Earth, or a melanin-like compound, which is then transformed into melanin precisely with that ultraviolet radiation that you mentioned. So the production of early compounds important for the origin of life is still a very key factor in this paper and in general in the approach that people that study the origin of life have. And the production of melanin and other compounds is helpful, because not only does melanin help mitigate the higher UV fluence that you mentioned that exists without ozone, where the protocells would be very susceptible to it, especially if they are on the surface. If we're talking about life in the subsurface, the radiation is not a problem. But if melanin and all of these other compounds are able to reach the bottom of the ocean, which we know that amino acids and sugars and all of those, if they're formed with UV, it would have been on the surface. If life formed below the surface, then melanin could have reached it. And as I showed you with my growth chart, it also has very, very good antioxidant properties and cell stabilization properties, even when it's not produced by the organism, so on the outside. And that was the biggest advantage that I saw, and I think is relevant, but because it was already well known that melanin is great at blocking UV radiation, not so much for X-rays, but still good. But particularly the effect that it has on cell health is relevant, because as they mentioned in this paper, it's very widespread on the tree of life. A lot of organisms in different kingdoms, in different families have melanin, and this points very strongly to an early appearance of melanin in the production of the cells. And this, of course, indicates that melanin would have been relevant and around the organisms at that time. And that's why I focused on it, not so much on the pigment radiation protection perspective, because if we're talking about the highest doses on these exoplanets, no pigment can protect them. It's more when we talk about what if it's in the subsurface or below a layer of water, is it still helpful? Can it mitigate the effect of some radiation, but above all, contribute to its cell health and protection from hazards in the environment?
Okay. Yes, I don't know.
Thanks for the talk. I have two questions. So one is about clarifying one of the last graphs that you were showing when you were having the different solutions, including melanin and seawater. So I did not get the why in the top panel. So why would, yeah, the previous, so go to the other slide.
Sorry.
Yeah.
This one?
Yeah, exactly. So why, when you have the solution that it's seawater, I understand, so 0.9% salt.
Yeah.
So why this is so low compared to the control sample?
Okay, because that was one of the other things that I wanted to test, because a lot of times when you're testing survival to radiation, people use these seawater solutions and they place the spores there and then they put them to grow. But that's not realistic at all when you compare to what an actual environment would be like. And so these solutions that are produced, they not only have the advantage of having the melanin here and no melanin here, but the organisms, they also excrete a lot of metabolites. They have some leftover nutrients from the solution itself, but they also release sugars, amino acids, a lot of metabolites that also can be used by the spores then as additional protection factors or for DNA repair. And so that's the difference that we see here. And I think that's the caveat you have with studying. Of course, you have the advantage of being studying only one factor when you study survival on saline solutions, but you also underestimate the survivability of real environments because when you have a lot of other metabolites, they survive better, but still with the melanin, it makes quite a lot of a difference.
Okay. Yeah, thanks. The second question was, I don't know if you have investigated in your experiments about putting radiation into the different populations of fungi that you have, variation in this event. So because the flares are things that are happening from time to time, then they don't happen. I don't know if you have done some experiment in which you let them grow without radiation, then suddenly you put for 1 day or 2 days a lot of radiation, then you stop the radiation, you have it. I don't know if you could see some kind of destruction of the colony that has formed or not, or if it just continues growing.
No, I didn't do it directly with the same colony, shall we say. That would be really interesting to do, even to see if there is an kind of acclimation or eventually genetic adaptation to such a high radiation environment. What I did do was see its constant growth after being exposed. So just letting it grow a bit, exposing and seeing it again just once, and there is a recovery, but it's not a full recovery. You can see it a little bit here. It's not the same experiment, but essentially what you see is that once the cells are irradiated, they never reach. So this eventually reaches a plateau, which is lower than the control one. So they will never reach the same concentration. If I was working with bacteria, probably or most likely they would, or at least be closer. But in this case, since they form very complex colonies and filaments, it's a really intricate process that is heavily damaged by radiation.
Thank you.
I'm not an expert in this field, but what would be the eureka moment for the astrobiologist? Like, what discovery would make you confident that, okay, some sort of life exists on this system? And is it possible with the current technology to have that?
In the solar system, yes. But it would most likely need an actual confirmation, for example, as the Mars sample return mission, which hopefully will be completed in the next 20 years or so. If you look at the samples and you determine beyond any reasonable doubt that there is something there, yes. Outside of the solar system, I think we're getting there, but it's really hard. Every once in a while, you hear sensationalist headlines. There was a recent one about K2-18b saying DMS was discovered, and not only was it not discovered, even if it was, it's not, one compound is not a biosignature. We also saw that with the whole phosphine debacle in Venus, and there's a lot of, every time someone uses one molecule or one potential detection to say that there is definitely life or most likely life there, you lose a bit of confidence in that actual work. So I think we need to be very careful when we say categorically that we found life somewhere else outside of the solar system. It's much harder, and I think to be 100% sure, it will take longer unless there's a big sign when we compare to places we can reach in the solar system.
Fantastic, Prof. You have been comparing UV and X-rays. There is a third flares also radiate protons.
Mhm.
How do you see the radiation of protons?
As I said, I did do irradiation. I think I have. I didn't do protons, but I did heavy ions. Oh, the screen sharing solves it because I have. Yes. Okay. Sorry. And now to share the screen. Okay, so these were some samples that I sent to HIMEC, which is a radiation facility in Japan, and we didn't use protons, but we used helium, argon, and iron nuclei. And what we saw was that there is a big hit on survivability, both for the wild type and the mutant. Essentially, similarly, these effects are not statistically relevant. Because melanin does not protect against the heavy ions, and they make quite a bit of damage. There is some improvement in their growth after that, but this is just their survival. And argon really limited their survival. So protons are similar to that. In general, the heavier the ion, the more the damage it does, but there are also less heavy ions from cosmic rays when compared to large amounts of protons. Proton flux is a very big problem, also due to the interaction with the atmosphere and atmosphere ablation. I didn't look into it directly, but it's definitely on par with UV or X-rays when we talked about a planet without an atmosphere. And that's why I also researched the subsurface, because in these cases, in the surface, it's very unlikely that something would survive.
Okay.
Yeah, well, thank you very much. Well, with respect to the energetic events like from stars or from galaxies, this is not my topic. So anyway, but just to say that there are some clear evidence that this energetic episodes are an opportunity from a chemical point of view, especially for probiotics process. Which, there are very important studies that reveal that we can jump from simple organic molecules like formamida to a sugar, to an amino acid or simple dust. And so, I mean, for us, I mean, what I want to say that for life, for us, are a problem, but for life origin, for probiotic physiology, perhaps it's opportunity.
Yeah, I think
it's at least this dilemma.
Yeah, exactly. It's a balance. So with my research, I don't mean to say that we have to be protected or life has to be protected from all radiation in a prebiotic scenario, especially the opposite, because a lot of researchers are starting to dismiss M-dwarf planets as potential oases for life because of the high radiation environments. And what I argue is exactly what you're saying, is that under the right conditions, there definitely can be some kind of attenuation of the radiation, but still a lot of it available to drive prebiotic processes like the production of bases, amino acids, besides the delivery from meteorites, and even the production of melanin itself, as I mentioned, is catalyzed by UV radiation. So even in a context where we're trying to protect the organisms or see how they survive high radiation environments, without any of this energy, it's harder for the origin of life to occur. And that's also what is a lot discussed when we speak about the icy moons in the solar system. So if there is no radiation from the sun reaching the depths of those seas, only with hydrothermal energy some of these reactions be catalyzed, but it's harder to do so. So I definitely think that there's opportunity there, like you mentioned.
Radiation together to minerals.
Of course, that's why we speak about the
That again, the
hydrothermal.
geology is very important.
Yeah.
But
No.
we always forget the geology. Anyway.
No, it's essential because the materials have come to somewhere, and the cycling, and that's why hydrothermal vents are, as far as I know, one of the most considered options for the origin of life nowadays, because it's extremely rich in minerals and in energy, which is absolutely key for the origin of life.
I have a question myself. It's probably beyond the work you've done, so probably there is no answer yet. But which is appearance of life versus survival, because you the work you presented, and most of the times we read about, it points to survival being something relatively easy, in the sense there are more and more strategies and means that the life finds to survive, even in very hard environments. But in order to have life, you have to first appear. Probably we don't know the answer, but do you have any opinion about how likely is life to appear under this kind of environment?
I mean, that's an impossible question to answer. My research definitely is not on the origin of life, it's on its survival. I try to mimic with the melanin solution and all of that conditions that may have been similar to the origin of life, but it's not my goal. But to your question, I think I think there are a lot of places in the universe where life could appear, just due to the sheer statistical probability with such a large amount of exoplanets estimated. But I feel like it's more unlikely than people think, and a lot of specific conditions have to be met. But maybe we don't know how life originated on Earth, and until we replicate it, which is still a work in progress, we can't really understand specifically how life originates, and even defining life is not so straightforward. A lot of biologists, or most, I would say, I think that viruses shouldn't be considered life, or don't consider them life. I'm not so sure. If we find a virus on Mars that's not from Earth, what does that mean? So there's also that point, but I think a lot of conditions have to be met, and that's not so easy.
Thank you. Any questions? Final questions? No? Okay, and that's send us off again.
Discussion. There is a discussion with this board. It's life or not. It's almost the same like the Bible.
You can stop the recording. You can stop the recording.